Welcome, everyone. We are so glad you are here to join us today. This is the first session in our professional track at the 2021 MDF Annual Conference. We are here today to learn about repeat instability and stabilization. As a reminder, this session is being recorded and will be available after viewing of the conference in the MDF Digital Academy at myotonic.org/digital-academy. I'm Tanya Stevenson, the CEO at MDF, and I am delighted to introduce our presenters today. To ask our presenters any questions, please use the chat function in Zoom at the bottom of the window. Questions will be answered at the end of the session. Okay, it's my pleasure and privilege to introduce our esteemed panel of experts this morning. Dr. Daryn Moncton is the professor of human genetics at the University of Glasgow. After graduating from the University of Bath with a Bachelor's of Science in Biochemistry, he went on to complete his PhD at the University of Leicester, investigating genetic instability at the mini-satellite repeat loci used in DNA profiling. He then received an MDA fellowship to move to Baylor College of Medicine, where he first began working on DM1. After completing his fellowship at the MD Anderson Cancer Center, he moved to the University of Glasgow, where he established his own group. He's internationally recognized for his work in understanding the molecular turnover and role of tandemly repeated DNA sequences in the human genome and their relationship to inherited disease, with a specific focus on the CTG repeat within the gene associated with DM1. Over the past 15 years, Dr. Monckton has contributed to numerous publications on genetic instability, has been awarded numerous grants, and is a sought-after presenter at myotonic dystrophy-focused conferences around the world. He serves on many advisory boards and committees, including the Myotonic Dystrophy Foundation's very own Scientific Advisory Council. Our next speaker will be Dr. Peter Bialik. He joined Triplet Therapeutics in 2018 as the head of translational sciences. He has worked in drug development for over 15 years, focusing on translational biology and biomarker strategies for early development into the clinic. His broad experience across multiple rare diseases includes neuromuscular, pulmonary, neuroscience, blood, and liver diseases. During this time, he's gained experience and insights in developing biologics, nucleic acid therapeutics, as well as small molecules. Before joining Triplet, Peter was the director of translational science at Intellia Therapeutics, where he worked across therapeutic areas to investigate appropriate biomarker strategies for target engagement, disease mechanism, and safety readouts. Previously, he worked at proteostasis, raw RNA therapeutics, and Pfizer. Peter received his PhD in biochemistry from the University of Western Ontario, Canada. Dr. Christopher Pearson received his PhD from McGill University Cancer Center, studying DNA replication and protein cruciform DNA interactions. Then he did a postdoctoral fellowship in the Texas Medical Center in Houston to elucidate the mechanism of disease-causing DNA repeat instability in myotonic dystrophy, spinocerebellar ataxia, Huntington's disease, Fragile X, and ALS. In 1998, he was appointed to the Hospital for Sick Children Research Institute in the program of genetics and genome biology, and cross-appointed to the University of Toronto in the Department of Medical Genetics, advancing to senior scientist and full professor. In 1999, he was appointed as a scholar of the Medical Research Council of Canada. He's been a member of the International Myotonic Dystrophy Consortium since its inception in 1999 and has served on advisory boards for the National Fragile X Foundation, National Ataxia Foundation, Hereditary Disease Foundation, Canadian Neuromuscular Disease Registry, BioMarin, GlaxoSmithKline, AOMO Pharma, and has served as an ad hoc scientific advisor for the fabulous Marigold Foundation, Treat and MD, and the Fragile X Research Foundation Canada. He has diligently served the American Society of Human Genetics and been a member of the awards committee for the American Society of Human Genetics since. Dr. Pearson's laboratory for the last 2 decades has studied mechanisms of disease-causing repeat instability associated with neurological, neurodegenerative, and neuromuscular disease, with the goal of developing effective treatments. In addition to a focus on how DNA repair can drive disease-causing repeat mutation, his lab also studies how DNA repair can protect against cancer-causing mutations. He has published over 80 peer-reviewed studies. It is my great pleasure to hand this over to Dr. Moncton.
Great, that's fantastic, Tanya. Thanks very much for that introduction and for introducing this session. So as Tanya said, I'm Daryn Moncton, Professor of Human Genetics at the University of Glasgow, and it's really great to be here today for this meeting and to talk to you a little bit about somatic expansion in myotonic dystrophy type 1. So what I'm going to do is tell you a little bit about the dynamics and consequences of repeat expansion in myotonic dystrophy type 1 and the therapeutic opportunities that kind of raises, and that will lead directly into the talks that Peter and Christopher are going to talk about. Well, they'll probably tell you a little bit more about that as well, but actually tell you much more about how we're going to translate some of these basic science insights, hopefully into new treatments for DM1 and related disorders. It's also on the bottom there, just my disclosures, just to make sure you're all aware of those. So as I'm sure we're all familiar with, the mutation in myotonic dystrophy is the expansion of a CTG repeat, and a key observation is that the number of repeats inherited is a major predictor of the severity of the symptoms and is inversely associated with the age of onset. So essentially, the more repeats an individual inherits, the worse the symptoms and the earlier the disease starts. We also know that the number of repeats is genetically unstable and that it increases from one generation to the next. And it nearly always gets bigger, and such that the disease severity increases in successive generations and the age of onset decreases in successive generations, typically of about 20 to 30 years per generation. And this obviously underlies the anticipation that forms a major part of the myotonic dystrophy phenotype that we see in families. What we've also appreciated is that the repeat is also somatically unstable. And this just shows some data here, looking at repeat length variation in the blood DNA of a myotonic dystrophy patient using a method we developed called small pool PCR, where essentially we're amplifying the CTG repeat in relatively small amounts of DNA in multiple PCR, such that we can see the products from individual input molecules. So obviously myotonic dystrophy is an autosomal dominant disorder, so half the time we amplify the small non-disease associated allele, half the time we amplify the expanded disease associated allele. And what we find is that very often the repeat length that's present in any one cell differs from cell to cell. So we put in more DNA, we can now see more common variants and get a good appreciation of the overall degree of variation in a given sample. And obviously we put in a lot of DNA, we can get a broader view of the amount of variation present in a sample. And again, what we typically find is that the small non-disease associated allele is somatically stable, whereas the disease-associated allele is somatically unstable. And again, from these types of data and other animal model data as well, we're able to interpret these distributions that we see in the soma as that this lower boundary here that we often see is likely indicative of the progenitor allele. That was the allele inherited from the affected parent, and that this process is biased towards expansions, and that there actually can be some cells that gain actually many hundreds of repeats, at least in blood cells, from the number of repeats that was initially inherited. This process, again, we've known for a long time, is highly tissue specific. And in particular, we find that there are very large expansions in the affected tissues, so specifically in skeletal muscle, cardiac tissue, and brain. And there's some data here from our group showing again that the repeat size in muscle relative to blood in this patient was around about twice the size. But actually in many patients, the data here from Charles Thornton and Richard Moxley's group, again, showing that the repeat size in skeletal muscle is often thousands of repeats bigger than it is in blood DNA. So obviously an important question is, when does this instability arise? And this shows some data here that we generated as part of the optimistic study. And what this shows here is that we're looking at the amount of repeat variation in blood DNA in 4 different patients here, each patient being sampled at 2 different time points. So in this case, when the individual was 46 and 55, 26, 48, etc. And what we can see is that the average repeat size grows between those 2 time points here. However, the rate of growth and the amount of variation overall is also quite clearly also dependent on the size of the repeat. So these relatively small alleles, less than 100 repeats, expand relatively slowly, whereas these larger alleles expand much more rapidly with time. So quite clearly, this is a dynamic process that's going on throughout the lifetime of the individual, and is highly dependent, therefore, on both the number of repeats inherited and the age of the individual. So what that means is that by, again, getting data from lots of individuals, we can build a statistical model that explains how much variation that we see in a given individual, depending on how old they are and the number of repeats that they started with. And when we do that, we can then establish for any one individual their residual variation. So that's the extra variation that we see independent of how old they were and how many repeats they were. And that forms essentially a distribution centered around about 0 in the population. So a simple way to think about this is that basically what we do here is we split the population into 2 halves. So these individuals here are all individuals who are expanding more slowly than average. And these individuals here are all expanding more rapidly than average, given how old they were and the number of repeats they started with at the beginning time. The important point to note here is that although these people are expanding more slowly, they are still expanding their repeats. So nobody is contracting their repeats, everybody is expanding their repeats, just some are expanding faster than others. And what we've been able to show is that those individuals who are expanding their repeat more slowly than expected have a later age of onset than we would expect, again, given the number of repeats that they've inherited, and that those individuals who are expanding their repeat more rapidly get an earlier age of onset than we would expect, again, dependent on the number of repeats that they've inherited. So these data are directly implicating somatic expansion in the disease process. And one other thing that we and others have noted over the last few years is, although the vast majority of myotonic dystrophy patients inherit a pure CTG repeat expansion, where it's just an increase in the number of CTG repeats, a subset of individuals, probably around about 5 to 10%, have acquired what we call variant repeats or repeat interruptions, where there's a different sequence intermingled among the CTG repeats. And this is most often CCG repeats. Most often they're at this right-hand end of the repeat, but again, occasionally they're in the middle, and occasionally they're at the other end of the repeat. But usually it's at this end, and usually it's CCG. Occasionally it can be CGG, and again, there are a few other variants out there as well. And these are present in around about 5% of individuals. And it turns out that these variant repeats, again, can have a dramatic influence on both the intergenerational instability, the somatic instability, and also the phenotype of the disease. I'm just going to, again, tell you about one particular atypical Scottish family that we've characterized, where there were the father in this family, again, had typical myotonic dystrophy, typical onset, typical symptoms. One of his sons, again, had typical onset and symptoms and was much more severely affected than the father. Again, often what we see in intergenerational transmission of the mutation. However, his sibling here had an ambiguous genetic diagnosis. It wasn't clear whether she really did contain an expansion, and she had no apparent symptoms of the disease. So we investigated this family and determined that actually the father and the son with the typical presentation of the disease had pure CTG repeat expansions, and that number of repeats had gone up from father to son, consistent with the anticipation that we saw here. However, the daughter had acquired de novo these variant repeats, so they weren't present in the father, but they were present in the daughter. She had this big chunk of a sequence here where we actually now have a hexamer of CTG repeated around about 50 times in the middle of this allele. And again, So that again explained partially why she had an unusual molecular diagnosis. She really does have an expansion, but it's not a pure repeat expansion. But it also appears to explain why she's much more mildly affected. In fact, she has very minimal myotonic dystrophy symptoms. It's almost certainly the case in that unless other people in this family had more severe symptoms, there's no way that she would have ever known she has myotonic dystrophy, at least in this stage. And she's now at least 20 years beyond the age at which she would have expected to have been have overt detectable myotonic dystrophy symptoms. So quite clearly then, the presence of these variant repeats can give rise to unusual inheritance patterns and then also unusual clinical presentations in a given individual. And again, one of the things that we've noted is that these variant repeats are associated with much more stable repeats in the soma as well. So again, this shows an individual here who's inherited around about probably about 250 repeats, sampled at age 16 and 23. You can see even at age 16, there's a lot of variation. And at age 23, there's now much more variation, repeats getting bigger on average and much bigger in a subset of cells. Whereas if we look at this individual here, inherited a similar number of repeats, but even at age 60, there's relatively little variation. And from age 60 to 70, again, only a very small increase in the number of repeats. And it's this stabilization of the repeat that we believe is why these individuals are much less severely affected. Again, both of these phenotypes or these observations are replicated in larger cohorts here. Again, I'm showing you data from the optimistic cohort, but we've seen similar observations in multiple different DM1 populations now. And that is that the presence of these variant repeats here, as indicated in blue, leads to less somatic instability and a delay in the onset of symptoms here. So again, these individuals with variant repeats have an onset that's typically 10 to 20 years later than we would see in individuals inheriting a pure CTG repeat. Again, this impacts not only the absolute age of onset, but also the many different individual progressive phenotypes that we see in myotonic dystrophy, as illustrated here in terms of pinch strength, so the ability of an individual to pinch tightly between their forefinger and thumb. And again, that's something that again is dependent on the number of repeats inherited. So individuals inheriting longer repeats generally lose their pinch strength quicker. However, those individuals with variant repeats lose their pinch strength much more slowly, again, suggesting that this is affecting not just age of onset, but the absolute rate of disease progression across probably multiple symptomatic domains. And again, some additional data here, again, showing similar to the data I alluded to earlier on, this is now individual variation in pinch strength corrected for the number of repeats inherited in the age of the individual at a given time versus the residual variation in somatic expansion. So again, people here are the people who are expanding more rapidly and have a greater decrease in their pinch strength versus these individuals up here who are expanding more slowly on average, and again, are preserving their pinch strength longer. So again, very strong correlation here between somatic instability and individual phenotypes that we see in myotonic dystrophy. So these data, therefore, are strongly suggesting that somatic expansion is contributing towards the disease process. And that therefore it might be a therapeutic target. So obviously, if we're thinking about targeting this and preventing repeat expansion, we need to know how that is mediated. And through a massive amount of work, both by our group and multiple other groups, including Christopher's and many other groups throughout the world, working not just on myotonic, but on many other related disorders that have a similar genetic mechanism, it's now very clear that repeat instability and somatic expansion is driven by the aberrant activity of a number of different DNA repair proteins, primarily from the mismatch repair complex. And in particular, this MSH3 protein appears to be critical for generating and driving somatic expansions, and therefore is a potential therapeutic target. And as well as lots of animal model data, what we have been able to show is that also natural common variation in the MSH3 gene. Again, these are not cancer associated mutations in MSH3. These are common, naturally occurring polymorphisms that probably only relatively subtly affect the activity of the MSH3 gene, are associated with variation in both disease severity and the rate of somatic expansion. And again, this is just illustrating here some of the polymorphisms that are present in exon 1 of MSH3 that we've been able to link with variation in both somatic expansion and age of onset. And notably, and kind of bizarrely in a way, there's a 9 base pair coding repeat polymorphism in MSH3 that varies in both the number and exact sequence of those variants. And the 3 repeat allele is the one that's most common in the population. And we've shown that, again, the more copies of that an individual inherits, the less the rate of repeat expansion, and the later their relative disease onset. It's showing that that's a protective allele relative to these other longer alleles in the population. However, it's fair to point out that this 9 base pair repeat polymorphism is in linkage equilibrium with a number of other polymorphisms in this region, and we don't know for sure whether this actually this coding polymorphism is functionally relevant, because there's also data that shows that these alleles are also associated with lower expression of MSH3. And it's probably that lower expression that is at least in part driving this association. But nonetheless, these are key data because they really illustrate that naturally occurring polymorphisms that are non-pathogenic in the human population can have a significant influence on both the somatic expansion and therefore the knock-on consequence on disease severity in this disorder. So what we now know is that longer repeats cause more severe disease. Intergenerational repeat length expansions cause anticipation, and everyone's kind of really recognized that for a long time. But what we now also know is that the repeat length is also increasing over time in somatic tissues, and that very large somatic expansions accrue in the affected tissues. And now we've got this direct data that individual specific variation in somatic expansion correlates with individual variation in disease severity, and that these variant repeats are reducing both somatic expansion and disease severity. And these candidate gene studies that are linking DNA repair with somatic expansion and disease severity all really suggest that somatic expansion is a key target, and that these DNA repair proteins that are involved in that may actually be the protein targets for new therapeutics in DM1. So it kind of gives us a new way to think about the disease. In the past, we've just thought about it as a CTG repeat that generates toxic CUG repeat at the RNA level, and we should be thinking about kind of directing our therapeutics here to try and alleviate disease. Whereas what I think we should be thinking about now is a bit more sophisticated way about thinking about this disease, is that we have a given number of repeats that's inherited, but actually what is truly pathogenic is the consequence of somatic expansion, generating a much larger CTG repeat, generating a much larger toxic RNA molecule that then generates the disease. And obviously that this process of somatic expansion is key to this disease pathway, and that's driven obviously by the number of repeats inherited, the age of the individual, different rates of expansion in different tissues, that these variant repeats can actually modulate that process, reduce the rate of expansion, and that these polymorphisms in these DNA repair genes can also influence that process. But what I think we can also imagine is that we could also have a drug-induced intervention here, again, that was to reduce the rate of somatic expansion, and we'd end up with a smaller CTG repeat in the affected tissues, a smaller toxic CUG molecule that would therefore induce a lower degree of disease in a given individual if we were able to modulate therapeutically this process. And again, I think we're at the point now where the data are very strongly suggestive that if we were able to do this, we would definitely have a very positive therapeutic impact. Obviously, ideally, what we'd like to do is be able to shrink the repeats, and Christopher may be able to tell us a bit more about how we're able to do that, and Peter may tell us a bit more about how we modulate this process here. Again, one thing I think we don't yet understand, which I think is still a key question, is as to whether somatic expansion is absolutely required to generate the phenotype. So we know probably, at least in the case of congenitally affected individuals that already inherit a very large repeat, almost certainly somatic expansion is not required, at least for those initial symptoms. It may still be required for the symptoms that we see later in the disease course. But I think particularly the data with the variant repeats strongly suggests that somatic expansion may actually be absolutely required to generate any symptoms in this disorder. But again, I think that's yet to be determined. And just finally, wanted to make one more point. I've obviously talked to you about the data here from DM1 that I think are very compelling. Again, we know much less about somatic instability in DM2, but it definitely is there. And I'd be very surprised if essentially a parallel process is not operating in DM2 as well. And actually, we've now learned a lot from essentially replicating all of these observations in Huntington's disease, and again, see essentially exactly the same effect in Huntington's disease as we're seeing in myotonic dystrophy. And also, again, some recent data implicating MSH3 also in X-linked dystonia Parkinsonism. And again, I think it's very likely that this somatic instability via similar pathways is operating across multiple disorders. And that's obviously attractive in terms of the fact that we can learn about DM1 by insights into these other disorders, as well as the other way around as well. And also, again, as Peter will, I'm sure, allude to, the idea that a drug that we develop for one of these disorders to impact on somatic instability may be beneficial for all of these different disorders. And that's obviously great for the individual disorders, but also is a major driver for the pharmaceutical industry in being able to tackle multiple disorders with potentially a single drug. So I shall leave it there, not first of all, however, without thanking the members of my group who've contributed to the work we've been doing, not just in myotonic dystrophy, but in Huntington's disease and related disorders, but particularly in terms of the work I presented today, Gail, Sarah, and Vilja, with the work we've been doing on DM1, and also our many collaborators in Scotland, Cynthia and Jean-Mathieu and the group in Canada, and our optimistic collaborators across Europe, and Fernando in San Jose, again, really contributed to the work we've been able to do on myotonic dystrophy. And obviously, absolutely critical to that is all the patients and families that have contributed their samples and data. Really grateful to those, and obviously the funders of our work, and in particular our myotonic work over the last few years has been funded by Muscular Dystrophy UK, the Myotonic Dystrophy Support Group, MDA, and the CSO. And I'll leave it there. As Tanya said, we will have questions at the end of the session. So I will now stop and pass over to Peter, and he will hopefully tell you what Triplet may be able to do about this. Thank you.
Great. Thank you very much, Darren. And I want to thank you very much for the organizer for giving us this opportunity to present the triplets strategy to treat repeat expansion disorders. I'm really excited to be here as part of the session, and Darren's done an amazing job of sort of setting the groundwork, and hopefully we won't have to go through some of the background again. Well, what I'm hoping to do today, and this is a disclaimer, I am an employee at Triplet. What I hope to do today is show you how we're taking some of those concepts that Darren mentioned earlier and really start developing drugs that can not only treat HD or myotonic dystrophy, but really many other repeat expansions disorders. So this is the brief outline for today. We'll go over a few key details that Darren mentioned earlier about somatic instability and the role of these mismatch repair, or otherwise known as DNA damage repair genes as well. Talk a little bit about why we selected MSH3 as our lead candidate going forward, because I think it's important to realize that not all genes are equal when we think about drug discovery. I'll show you some really interesting preclinical data that we have on a tool molecule that shows that we can block somatic instability with an MSH3 inhibitor. I'll share some really key data on how we hope to deliver 3360, our lead candidate, into the brains of HD patients, and then follow up at the very end about some clinical data that we're generating with Shield HD and how that influences our biomarker strategy. So as Darren mentioned earlier, the repeat expansion disorders that we often refer to as REDS, there's over 50 of these repeat expansion disorders that have been identified today, and more appearing on a daily basis even. They are characterized by an increased number of these specific DNA repetitions in specific genes. So in the case of Huntington's disease, for example, we have the CAG repeat length. In a normal allele, this is approximately 16 to 20 repeat lengths. But in a disease-causing allele, we know it's closer to 36 and above. What's also been apparent, and Darren mentioned this quite well, is that just inheriting the disease allele length is not enough to derive symptom onset. It's enough to predispose you to the disease, but we believe there's this two-step process where you inherit the disease, but you need somatic instability to also occur in order for it to have full disease onset. This is illustrated in this cartoon here. And a couple of things to note. Originally, the dogma was that you were born with this inherited disease length, say 40 repeats in the CAG for the Huntington's disease, and that this was stable over the entire lifespan of the individual. We now know that it's not the case, and Darren showed good examples of that in muscle and blood. For Huntington's disease, it is quite prominent in the brain, in multiple different brain regions. As an HD individual, if they're born with that 40 repeat length, that state is not stable. It is unstable and begins to increase in the number of lengths in the HTT gene. At some point, which we have very poorly defined, there is a sort of a threshold that we think about with these repeat lengths. When cells and neurons cross that length, they start becoming dysfunctional. And if you get many of these neurons that have these increased CAG repeat lengths, those actually leads to neurodegeneration. You start seeing brain loss, you start seeing symptoms appearing in those patients, as is decline, executive function, motor manifestations as well. And very much late in the disease, there's full HD onset. What we're deciding to do in Triplet is to really focus on these early stages of the disease where we can hope to block somatic instability and prevent it from going forward. And you saw in the earlier talk, there's good evidence to support that. You saw these blots earlier for myotonic patients, and these are worth seeing. This is occurring in HD patients as well. One thing to note for HD, we do know that the amount of somatic instability takes place even many years earlier than the age of onset. And in these two individuals exemplify that. And in the case on the 13 years, there is each of these individual dots exemplifies that there is a neuron that has an increased number of these CAG repeat lengths, but it's not present in the cortex at this time. Is when you get to another individual who's much closer to that age of onset, where you see these increased CAG repeat lengths in both the striatum and cortex. Again, these are two areas that are particularly prominent and important for Huntington's disease progression. Darren also alluded to earlier that the CAG repeat length can indicate when you are going to exhibit symptoms. But what's also interesting that has come out of some of these studies is that you can have an individual that has, for example, a repeat length of 40. And in one case, that individual has a very slow progression and has a very late age of onset. A similar patient that has repeats length of 40 could also have a very rapid progression and show much earlier age of onset. And this led to GWAS studies identifying these mismatch repair or DNA damage repair genes as a class of genes that seem to be affecting this process. What's interesting to know is that there are SNPs or mutations in and around the MSH3 gene that if you lower the expression of MSH3, this actually slows the progression, and these individuals tend to have a later age of onset. Conversely, if you have mutations or SNPs in MSH3, which stabilizes the protein, this has the opposite effect, so that you have a much earlier age of onset and a rapid progression. So this link between MSH3, somatic instability, and disease progression is quite strong in the HD community. And this gave us the impetus to start the company Triplet Therapeutics and really develop a MSH3 lowering oligonucleotide therapy. And I'll go to that in some extent in the next couple of slides. But we believe that by blocking somatic instability, by modulating MSH3 expression, we can prevent further somatic instability from taking place, and this will have a positive impact on disease progression. There's a few things that are different from Triplet's approach compared to other, say, HTT lowering approaches, and there may be similar analogies in the DM1 field as well. With Triplet Therapeutics, we are really targeting what we believe is the upstream event. We want to block somatic instability from taking place, and this would prevent any further expansion of this disease allele from taking place. This is very different from some of the HTT lowering therapies, which would potentially decrease both the wild type allele as well as the mutant allele as well, and that could have deleterious effects. We also know from various animal studies, and I'll show you one in a second, that we only need to achieve about 50% reduction in MSH3 to have a positive impact. And this actually has a really profound impact for drug discovery, because we don't need to achieve those high levels of like 90 or 95%, which you would typically expect from drug discovery and thinking of like, say, a kinase inhibitor for cancer. We don't need to hit the target that hard. Also, it's really important to note that this mechanism, we believe, is conserved across multiple DNA of these reds. And as such, if we can develop a therapy for Huntington's disease and show it's safe, we have the potential to have a positive impact for other CNS repeat expansion disorders as well. So why is MSH3 such a good target? When we first did our due diligence looking at all the mismatch repair genes, MSH3 clearly came to the top very quickly. When you look at the NOMAD database, and this really looks at all the genetic sort of variations in a large number of healthy individuals, we saw that MSH3 loss mutations are relatively tolerated amongst the general population, meaning that if you lose one copy of MSH3, you really do not see any phenotypic disease sort of causing alleles. This is different than, say, some of the other genes in the mismatch repair genes, which are related to colorectal cancer as well as Lynch's syndrome. Another key thing here is that when you look at the MSH3 knockout mice, these mice have a normal phenotype. There's no overall phenotype, they have a normal lifespan. And this is really important for us as well, because it suggests that there is some functional redundancy potentially with other members of the mismatch repair genes that compensate for this function. And in those knockout animals, they didn't show any predisposition to cancer, which is important as well. We took a look at the MSH3 gene and designed and looked at in silico analysis. So we tried to pick oligos which had very good particular biophysical characteristics. We screened approximately about 500 of those ASOs in vitro cell culture models. We took a subset of those through in vivo rat tolerability. We analyzed them in non-human primate. Eventually, we came up with our TTX-3360, which we've nominated and is currently in an IND-enabling studies. Unfortunately, 3360 does not cross-react with a mouse, and we were not able to use this as a proof of concept in a mouse model. But what we were able to do when we were doing the screening is identify one molecule in particular. We call this our tool molecule 3281. This does cross-react with both mouse, human, and non-human primates. And we were able to evaluate its efficacy and effect on somatic instability in two different HD mouse models, the Q111 and the R62 mouse models. The Q111 is relatively mild and resembles more of an adult onset for HD. The R62 is much more rapid and severe. It resembles more of a juvenile. In both cases, if you administer our 3281 to the ICV administration to the brains of these mice, we saw in both the striatum and the cortex regions about 40 to 60% knockdown of the MSH3 gene. And when you look at and measure the somatic instability, and this is we're referring to here as a somatic instability index, we saw a dramatic reduction and almost complete stopping of somatic instability in both the striatum and the cortex. Again, this is really important data for us, since it really suggests in the clinic, we really should only look for 40 to 60% knockdown of MSH3, because the data here with the mouse models, as well as the genetic data that's being presented already, really shows that you don't need to hit this target 100%. And again, that has really good biological implications as well. When you think about intrathecal administration to get to the brain, this is a strategy that has been used by Spinraza to deliver the SMA drug, as well as tominersen for HD as well. And in both those cases, they used the intrathecal administration. We wanted to evaluate whether this would be a good way to get to the deep regions of the brain. For Huntington's disease, we want to get to this sort of light colored area here. This is the sort of the striatum and the deep areas of the brain. But when we delivered a single injection of our drug into the non-human primate via the IT, we didn't see really good exposure in this brain area here. When we looked and used a different route of administration, this is using an ICV type approach where we're directly injecting our oligo into the brains of these animals, we saw much higher distribution to this area. And this gave us confidence that if we really wanted to target the regions which are very susceptible to somatic instability, that show very early changes in brain volume, this is where we need to be. This is the route of administration that we have preferred. Taking this information, we performed another longitudinal analysis of a very complicated study. We took non-human primates, gave them a single injection of our TTX-3360, our lead candidate, and then looked out to look at the durability as well as the efficacy of our treatment in these animals, all the way to day 84. We looked at a number of different brain regions at necropsy. The caudate, putamen, and nuclear accumbens are subregions of the striatum. We also looked at the frontal cortex, the temporal cortex, as well as the cerebellum. If we focus initially just on the frontal cortex, even we saw as early as day 29, so 4 weeks after that initial injection, it's a very good rapid knockdown of our MSH3 target in both the ipsilateral and contralateral sides of the brain. So the contralateral being the opposite side of the brain that we've injected into for the non-human primates. And you can see in these two tissues that this is knockdown is sustained all the way out to that 12-week time point. In the caudate and putamen, we also saw very good knockdown, and we did achieve that 50% knockdown that we were looking for. In the case of the putamen, it is a little bit harder of a tissue to get to. It's more encapsulated a little bit, so it has some white matter that the oligo needs to travel through. But we did achieve that 50% knockdown, even maintaining that levels almost out to day 84. Again, this is really great data because it suggests that just a single injection can have a sustained and powerful knockdown effect on MSH3. This has potentially really important implications for dose level as well as dose frequency when we get into the non-human primate IND enabling studies, as well as potentially dose regimens for clinical application as well. It's really, really powerful and important information to know. One of the things that we know and recognize about Huntington's disease is that it's very complicated. There are those early phases that I mentioned to earlier, which show some symptoms, but not others. We really want to, again, focus on this early stage of disease progression for Huntington's disease. As such, we actually designed our own natural history study to collect data to allow us to understand the disease progression early on for Huntington's disease. We enrolled 70 patients, approximately over 6 months, into 3 particular cohorts. Cohort 1 is really the prodromal, so they have that repeat length, but are really not showing any manifestation of any symptoms. Cohort 2 is beginning to exhibit some of the symptoms, and cohort 3 is more what we refer to as sort of manifest patients. We've collected 3 basic different types of information. We're looking at volumetric changes, so we're looking at MRI to see what are the changes in the brain matter that are occurring in this HD patient population over a 2-year period of time. We're also collecting motor functional scores as well as sort of executive and behavioral scores with the UHDRS. We've also included Csf Sampling Collection, and I'll get to this in a second. This is really important because it's going to enable some of our biomarker studies going forward. We're currently over a year in collecting data for some of these patients, and we're hopefully will be able to share some of this data in the near future with you. But it's really exciting for us to be able to get a more comprehensive view of these early stages of HD disease progression. So why are those CSF collections really important for us? Well, first, it really helps us enable our translational biomarker strategy, in which there are 3 main parts. One is to be able to characterize the disease and be able to combine both the clinical outcome measurements, the biofluid collections, as well as the neuroimaging. In the biofluids, we know that genes or proteins such as NFL, mutant HTT are increasing, suggesting that there's disease progression going on. Target engagement is a term that we often use in drug discovery. And essentially what it means is that you would like to know at a given dose that you are inhibiting MSH3 expression to the level that you expect. Again, this is really important for us to know going into the clinic to say dose X gives you Y effect on MSH3. We also want to be able to measure somatic expansion in our HD patient population, since our whole hypothesis is that lowering MSH3 will have an effect on somatic instability, and then this again would have an effect a positive effect on disease progression. Unfortunately, with these last two, target engagement and somatic expansion, we're not able to sample the patient's brain tissue directly. These patients are living, and we have to use a novel approach that will allow us to look at collecting these types of information. And we've employed a new novel biomarker strategy where we're using extracellular vesicles that can be found in the CSF. So a quick primer on extracellular vesicles, or we often refer to them as EVs. EVs are these lipid bilayer vesicles. They're approximately 40 to 200 nanometers in size. They are actively secreted by living cells. They're not secreted, for example, by dying cells. That is a completely different process, and they're not captured here. The EVs encapsulate a lot of biological relevant material. They contain RNA, protein, and metabolites. They do not really contain genomic DNA or fragments of genomic DNA. So something important to note, and I'll come back to that in a second. There really is no standardized methods to isolate these EVs or even isolate and understand what is in the contents of those EVs. So this is the first question that we ask. Can we even use these EVs that can be collected from CSF as a means to enable our biomarker work? So the middle panel here shows one example of an experiment that we recently did, where we took EVs from 5 individual patients. We collected those EVs. We extracted the RNA that we could from them using commercially available kits, and then performed RNA-seq. What we were really hoping to see, and we did, is that MSH3, which is our key target, can be detected in the EV's RNA. Also, HTT is also detected in the EV RNA. And this really enabled us to go forward and start designing experiments to look at target engagement and somatic instability. But an obvious question is, do the EV content actually represent what's going on in the parental cell that's secreting these EVs into the biological matrices? And the answer is yes. If you look at the HeLa experiment that we did on the right-hand side, it definitely does show that. We took HeLa cells and transfected them with our lead compound, 3360, and after 72 hours, we saw about a 70% reduction in MSH3 levels at the cellular level. When you look at the EVs that are secreted by these HeLa cells into the conditioned media and ask the same question, we also saw approximately a 70 to 80% knockdown or reduction in MSH3 expression in these EVs. So this is really, again, great news, because it suggests that whatever content, MSH3 content that we see and can detect and measure in these EVs, they represent what's going on in the parental cells that are secreting them. Again, this is really important data to enable our target engagement assays going forward. And we're currently looking at EVs from CSF in non-human primates to, again, draw that link between what is going on at the brain level, the MSH3 knockdown level, compared to what we see in the EVs. Last but not least, we are very much interested in being able to measure somatic instability as well. But I alluded to earlier that genomic DNA is not present in these EVs. So what can we use as a potential surrogate? And HTT RNA may be indeed that fact. To see if we can actually measure the CAG repeat length in the RNA, we took DNA and RNA from iPS cells derived from an HD patient. If you use the fragment analysis that is similar to what Darren showed, you can detect a modal length of 123 CAG repeats. When you isolate the RNA from those cells, you also saw and be able to measure a modal increase of 123 repeats. There is almost a complete direct correlation between the number of repeats in the DNA and the number of repeats in the RNA of HTT. And again, this gives us really important confidence that as we go in towards the clinic, we'll be able to potentially use HTT RNA in the EVs to measure somatic instability. So where are we now in terms of our drug discovery pipeline? We are currently doing our IND enabling studies for 3360 for HD, and we hope to submit an IND in the mid of 2022 next year. Our SHIELD HD study is continuing to provide powerful information for us, and we are using that information to design our best clinical phase 1 study. Those CSF collections, although I haven't been able to show you them, are really helping us as well to design that target engagement biomarker assay and sort of do confirmation studies before we get to the clinic. We recognize that our drug TT3360 can be applied for potentially multiple Rett diseases that have a CNS manifestation. And in particular, we are supporting the myotonic dystrophy type 1, the NDM1 natural history study, as well as one in DRPLA, because we recognize the need to understand these early disease stage progressions. If we have, or not if, but when we have good clinical safety data from our phase 1 HD trial, this will allow us to rapidly move into these other Rett indications. Hopefully, I've given you some really important information and shown how we're taking some of the genetic data and apply it into our drug discovery to not only treat HD, but potentially myotonic dystrophy and other repeat expansion disorders. So for the sake of time, I will stop here and pass the baton off to Christopher.
Thank you very much to both Darren and Peter for their wonderful presentations and setting up the background for me. So I'm going to be talking to you today about repeat instability with the goal of trying to actually reverse somatic expansions of the repeat. So I bring this slide up because I want to convey to you that repeat instability in myotonic dystrophy is really, really high compared to other diseases. And you have various disease forms, including the congenital form, which is unique to myotonic dystrophy relative to all other repeat expansion diseases. So it's in utero where the child is affected. And with increasing size, there's increasing severity. In all of my slides, I use the same color coding for the unaffected, the proto-mutation who has an expansion but yet to be affected, and the various stages of age of onset. So as Darren just mentioned, not all DM families show expansion biases, although most of them do show an expansion bias. Some of them show a contraction, and sometimes there's a contraction bias. And this would argue that there might be something that we can learn about these families, about contractions, where it's actually naturally occurring. Is there something that Mother Nature can teach us about how we might be able to reverse the disease progression by looking at naturally occurring contractions? So here's a family that we've been looking at, a 3-generation family, and clearly it's going from an expansion of 700 to 500 to 300. So it's actually traveling with the chromosome in this family multiple times. This would suggest that there is something genetic within this family that we might be able to learn. This family also shows reducing severity of the disease as the contraction occurs through the family, and this is obvious in multiple phenotypes. So what may be affecting this differential instability in transmissions? And I show this slide because basically there's very, very minor changes which actually can make quite a bit of diversity. So a lot of us look different, just like butterflies look different, and that's because there's minor changes in our genes as there are in butterflies. And so what kind of variants might we be thinking about? So some of the factors that we might be thinking about would be cis factors. That would be things that are in line with the DM repeat. And Darren has mentioned some of them, which would be repeat length. Larger repeats obviously are giving rise to more likelihood for expansions, and interruptions, non-repeat units. And these could actually give rise to stabilization or polarization of the repeat instability. Other things that we've noticed in CDM, congenital myotonic dystrophy, is DNA methylation of the flanking regions of the DMPK locus. And these might also affect instability. And trans factors might also play a role. And we're not just talking about the proteins themselves, we're talking about minor changes in those proteins that might actually make them more or less able to cause expansions or contractions or stability of the repeat. And some of those are shown here, and they've been mentioned already, MSH3, MLH1, et cetera. And there's a new player called FAN1. And basically, we've been looking at at some families with contractions, and we're sizing them with southern blots. And we've actually got a collection going, and we're very interested in building this collection because we'd like to look at all of these different aspects. We've also been looking at interruptions of the repeat. And so, for example, we, like others, have found that the contraction families have interruptions, and the interruptions are polarized in the repeat tract. And there are various types of repeat interruptions that are non-CTG repeat units. And these have also been observed by others. And so these seem to be correlated with the contractions. And so it's an important factor to look at for the repeat contraction. So that would be a cis type of element: repeat length, repeat purity, and we've also been looking at DNA methylation. And so far our analysis is telling us that the methylation is not, at least in blood, having an effect with repeat contraction. And this is a limited statement because we have to note that there is large differences in DNA methylation between different tissues. And when you're talking about transmission, you're talking about a very specific stage of development. So as I mentioned, we have a study that's ongoing, and I can share this flyer with you. If any families are willing to participate in this study to increase our ability to have samples to look at the stability of the DM1 repeat, I urge you to contact myself by phone, or Peter, or my technician, or myself by email. So somatic expansion rate can vary between people. And Darren mentioned this, but I really think that it varies quite a bit in myotonic dystrophy and other diseases between individuals, possibly even within the same family. And so what I'm wanting to do is to show you some stagings. So we know from studies that we've done with pre-implantation genetic diagnoses that there is instability occurring during the very early stages of development after the sperm and the egg get together. We've also looked at fetal tissues for expansions, and this is a southern blot, and I'm showing you the size of the repeat above each of the bands in the tissues of the same human fetus. And you'll notice that there's changes between tissues even at this early stage of development before birth. And the same is true with this other fetal termination. So it tells you that instability is occurring early, but it also tells you when you look at postmortem individuals that we can have very large expansions, oftentimes as much as 6,000 repeats between tissues of the same individual. So it's quite an uphill battle that we'll be fighting when we're trying to target repeat instability. But I think that it is a worthwhile battle, but I think that we need to have multiple approaches to this. So let's look at this scenario. So you have the sperm and the egg getting together, forming the developing embryo, and then there's the child at birth, and then there's aging through time. And so if you have a person who's inherited only non-expanded repeats, as time goes, the repeat is completely stable. So if you have somebody who's inherited a size that's within the late on stage size, and they go during development, there's some expansions over the course of their life up to 65 years, you will see that there are some expansions. Okay, so now remember, there are variations between individuals. So some people could have much more rapid expansions, giving rise to more severe disease progression. Alternately, some people might have very stable repeats, and these don't change, and this person might not get sick, even though they've inherited the same size. Now, if we're thinking about treating an individual for repeat instability, we could have various scenarios. And so I'm presenting these lightning bolts as different drugs. And in all cases, I'm suggesting that we treat at the age of 5 years. I don't think that this is the right age. I'm just making it as a schematic to help you understand. And all individuals go to 65 years. So this is the individual who has quite rapid expansions. Let's imagine we treat with the yellow lightning bolt, and this actually is treated at the age of 5. So instead of going up that high, we actually stabilize it, as in what has been done by Triplet Therapeutics for MSH3. They stabilize the repeat. So this individual will have disease occurring, but it will be stabilized from getting worse by the progression that would be offered by somatic expansions. So this is what it would be without treatment, and this is what it would be with treatment. So I think that if we had such a treatment, it would be a good thing. Alternately, if we have a different drug, we might actually induce contractions, and this would be considerably beneficial compared to this. So contractions would be better than stabilization. So I think that we should continue to look for new targets that actually would induce contractions. Other drugs might actually induce contractions at a faster rate, and that would be wonderful. So comparing these different approaches, obviously the larger contractions, provided they're safe, would be better. So there are multiple targets, and we've heard from Triplet Therapeutics targeting MSH3. We've also heard about other mismetric paraproteins, including MLH1 and FAN1. And we and others are looking at FAN1 right now. And there's also looking at the DNA itself. My lab has published a paper where we're targeting the DNA itself, and targeting the DNA can actually induce contractions, albeit they're small contractions, but they are actually inducible. This tells you that proof of principle, it can be done. Interestingly, the mode of action of NA, which is the ligand that binds to the DNA structure, can actually be processed by FAN1. It is not a surprise, and that's shown in this bioarchive paper, it's not a surprise that all of these factors, including the DNA structure, would interact together. So one of the things I think we need to be aware of is taking advantage of the genome sequencing and the bioinformatics, which is a time that science is at where we're able to do massive things. And in this paper, my colleagues, Ryan Yoon, showed that by looking at autism individuals, you could actually parse to see their genomes, to see if there's an association with expansions. And in fact, they found thousands of repeats that were expanded, including myotonic dystrophy individuals with expansions, and they are associated with autism. And one of the things that we did is we took the sequences from those individuals and we parsed them for those that have different kinds of expansions. And what I'm showing you here is for those that are actually parsed for CGG expansions. A lot of the DNA repair proteins that we've been talking about, they don't affect just CTG or CAG repeats, they also affect other repeats. And so we're parsing them here for CG repeats and looking at the sequence of those individuals at their genome, looking particularly at the sequence encoding FAN1, which is a DNA repair protein, we actually identified significant variants that associated with those ASD individuals who are associated with CGG expansions. And it's not a surprise to us, but it may be to others, that we actually identified some of the variants that were identified in the GWA for Huntington's for FAN1, which might suggest that this subset of variants might actually also be associated with other repeat instability diseases. So I remind you, one of the things that we'd like to do in our study is look at those different variants by sequencing the genomes of those individuals. So I'd like to thank the people who've worked on this. This has not been an isolated effort. There's a lot of collaborators who work, and this is mostly the DM1 group in my lab and other labs that are working on this. And there are others that are working on this. And I'd like to thank the fantastic support of all of the agencies that have been putting effort into support into my lab's work. And I'll take questions.
Thank you all. We have a few questions that have come in. And I'm not sure, I'll read these out loud and you can let me know who might be the best to answer this. But Ben is saying, thank you very much for the crucial research. He's a DM2 carrier and he would like to know what you would hypothesize to the effect of a restricted protein diet might be on repeat expansion. So prior to diagnosis, he found symptoms to be modulated significantly by dietary protein. More protein intake corresponded with greater myotonia and heart symptoms. Lower protein intake corresponded with fatigue, potential endocrine issues, but less myotonia and improved strength. Anyone have any thoughts on it?
I can start off. So I think it's a good point that environmental factors are likely to be important too. So we know that there's wide variation in the severity of the symptoms that we can't account for just by the number of repeats inherited. And we've talked a lot today about how certain genetic polymorphisms are likely major contributors to that inter-individual variation. But there's absolutely no doubt that there are going to be environmental modifiers that impact absolutely on overall disease severity. And I think it's plausible to think that there might also be some environmental modifiers that actually modify the rate of expansion. However, I am not aware and don't really have a good basis for hypothesizing what they might be. Peter or Christopher might want to be more speculative. Peter's shaking his head, but so I think it's a good point that there likely is environmental modifiers. And I think, definitely, I think as a field, we've probably not put enough effort into identifying environmental modifiers in general, not just in terms of repeat instability, but into disease in general. And I guess in a way, that what the research that's come out of Optimistic and a number of other studies now demonstrating that exercise obviously is beneficial. Again, that may be directly acting via the muscle strength, and almost certainly is acting via that way, but there may be other additional effects. But again, it's a good illustration that environmental impacts can also have an impact on overall disease progression.
Yeah.
And I'm sorry, I can't say more about more specific about the particular dietary modification suggested.
I'll just jump in and say that I agree with Darren. There's a lot more research that likely needs to be done. Definitely in the GWAS studies, there are genes and targets that have come out of it where we know little about their function, but it does suggest that they can impact disease progression as well as age of onset. Some of these with some research may point to a nutritional component, but as of right now, I don't think there's any genetic data, at least, that would support that at the moment.
I can say that there is good support for the suspicion that there would be an effect, environmental effects. For sure, the genome stability can be modified by environmental insults. And there was a study recently done for ALS, form that is caused by a repeat expansion. And this study actually found that lifestyle choices, which included dietary input, actually did have an effect on outcome. And it was done relative to before the onset of disease. And so I think that there is a good reason to go and look at these types of input, but I do not believe that there is any data that I am aware of, and I'm not an expert, that would suggest that dietary limitation would actually be beneficial. I don't know the answer to that.
Thank you. And I think we had a follow-up question by someone else to that, asking if there are any social factors that the decreasing repeat individuals share. Supersize or diet as examples.
Again, I don't think there's no direct data, but again, I think I would recommend everyone tries to maintain a balanced, healthy diet and do moderate exercise. It's good for everybody in the population, and I don't think there's any reason to believe that that's not similarly true for for individuals and families with myotonic dystrophy and other repeat expansion disorders.
Okay, thank you. So Phoebe has a question for Dr. Monckton. Are variant CTG repeats commonly or completely random, excuse me, or do you see inheritance patterns with this?
So what we seem to see is that they are, so as far as we can tell, they are largely absent in the general population. So they're not there. And Christopher alluded to these pre-mutations. So those are repeats that are bigger than we typically see in the general population, but can expand in subsequent generations to cause the disease. And we don't think the variant repeats are in those particular versions of the gene. And they don't appear to be present in the first generation. So typically we would see a mildly affected grandparent, adult onset, and then severely affected children. We tend not to see them in that first generation. They tend to appear in that second generation. And nearly all of the cases where they've occurred as a brand new event, it's from a transmitting father. So we've never seen a female passing these on. It's always fathers that pass them on again, or at least in the limited number of cases where we've seen them occurring de novo. So it's not to say that they could never occur by female transmission, but certainly there's a bias that they occur during male transmission.
Yes, so I'd like to point out that the contraction families are probably more common than we know from the literature. And that's probably because people go to the hospital when something's wrong and they don't go when something's not wrong. And so family members who are aware that they have myotonic dystrophy and they are seeing milder symptoms in their children would be helping the community, like what we're doing, looking at instability as a target by coming forward and sharing samples, or at least letting their doctor know that they have somebody in their family who's more mildly affected. And that way we would know how many more there are. And doing sequence analysis of the repeat purity and the methylation, et cetera, would actually benefit us in being able to get a handle on this. So yeah, but commonality, I don't know if there is something common between all of them that's outside of their genetics.
Thank you. We have another question. What is known about the mechanism of repeat expansion in post-mitotic tissue, like muscle and brain? So how might repeat expansion affect our common cell and animal models?
I could take that and maybe Darren could follow. So we've done a lot of work on the mechanisms of repeat instability, and post-mitotically, there is no genome duplication occurring. So that means the general term of DNA replication would be excluded, but that doesn't mean that DNA polymerases are not active during processes of DNA repair or recombination or so forth. Specifically, it has been shown by our lab and others' labs that transcription across the expanded repeat is definitely driving the instability process. If you have transcription on one or the other or both simultaneously, you can actually enhance the process of instability. And this is likely due to the fact that it's actually forming an unusual structure during the transcription process. And the processing of that structure, which we don't have a very good idea of what's happening, but we do know that DNA repair proteins are processing those structures to larger expansions. But you cannot imagine thinking of stopping transcription as being a beneficial thing if you're going to have a loss of the product. So you have to be very careful about how you attack this type of problem. So I believe that in many of the diseases, the repeat is being transcribed in the post-mitotic cells, including those that are showing the larger expansions. Earlier studies where they're looking at the levels of transcript as representative of transcription may not necessarily come to the right conclusions because they're looking at the levels, not the active process of transcription, right? So transcript levels could actually represent transcript stability as opposed to just the production of the transcript. And I suspect that it's the production of the transcript that would be the process that's driving instability.
We have a question from Peg. What is the relative contribution of EPOL and rate of somatic expansion to age of onset? So if you control for effect of EPOL on age of onset, what is the effect of rate of somatic expansion that is above and beyond the effect of EPOL?
So I'm going to first of all translate that. So I think EPOL is estimated progenitor allele length. So that's the number of repeats that someone has inherited. So it's quite clear that the number of repeats inherited is the primary driver of disease severity. Okay, if you only inherit 60 repeats, you will not get congenital DM1. And if you inherit 1,000 repeats, you will not have late onset DM1. You know, that's clearly the primary factor. So what I can say is that when we do those, when we correct for progenitor allele length and look at age of onset, the residual variation that we account for in blood is relatively modest. So it's only a few % of the variation in age of onset that's accounted for by variation in somatic instability in blood. And I think that's encouraging in that we are able to get some association. So it's telling us that there really is a cause and effect relationship there. However, what I think is we're underestimating the true effects because we're not actually measuring the rate of expansion in the key target tissues, which would be muscle or brain or cardiac tissue. And that's that's one of the pieces of data that we don't really have yet to kind of get that true impact on the link between somatic expansion and age of onset as mediated by the rate of somatic expansion in the truly affected tissues. But actually where we go and look directly at the effects of DNA repair genes, that kind of gives us a more direct link there. And again, we don't really have as good data in DM because we just don't have enough big, well-characterized clinical cohorts, but the data from Huntington's disease are really quite striking that, you know, overall now, if you add up all of the genetic modifiers that are modifying that we think are modifying the rate of somatic expansion, they're probably up now to accounting for 20 to 30% of the variation in age of onset. So that's a pretty substantial effect. We're not just tinkering around the edges here. These are major effects that we're having, or that somatic expansion is having.
Yeah, I want to add that some of the variants that have been identified, at least in HD, and I suspect that they're going to be true for other repeat diseases because a lot of these DNA repair proteins don't affect just myotonic dystrophy or Huntington's models. They also affect Fragile X. So these are different repeats altogether. And they can have effects where they dramatically delay or speed the disease stage by up to 6 years. And the purity of the repeat can delay age of onset by up to 29 years. So these are major players. And so identifying things that could give you a handle on treating this could actually delay disease by many years. So yeah, these are not small players. These are big players. I agree with Darren completely.
Thank you. We're a little over time at this point, but there is one last question, and if you have a moment just to answer before we end. In DM2, the correlation with expansion length is poor. Can you attribute this to the complex repeats? And is there any known clinical correlation?
I can touch on that a bit. So yes, there clearly is very, very high levels of somatic expansion in DM2. Such that there's no detectable association between the number of repeats that you measure in a given individual and the severity of the symptoms. But again, we have the same problem in myotonic dystrophy type 1 and in Huntington's disease, that in all of these disorders, we have, again, another example of an ascertainment bias, that we sample individuals in a way that's inversely proportional to their severity of their symptoms. So we sample mildly affected individuals when they're very old and severely affected individuals when they're very young. And that can mask some of the somatic expansion effects. So you can imagine with DM2 that individuals who inherited a smaller allele then were sampled, because that then was going to give rise to a later disease, which then they didn't get sampled till very much older, but then by the time they're very much older, their repeat has grown. So it might look like it's the same size as someone who had a more severe disease, who presented much earlier and were diagnosed much earlier, and we took the blood sample much earlier. So I think that's happening so rapidly in DM2 that we can't really say whether there really is an association between the number of repeats inherited and the severity of the symptoms. Kind of in a way we know there is, in that if you have less than a certain number of repeats, you definitely don't get the symptoms, but whether it's truly a threshold or a gradient, I think there's likely to be a gradient. We just don't have enough samples from people sampled early enough to really address that.
And maybe, Darren, I can just hop on quickly to comment something. And this is something that also Chris mentioned earlier, is if people are interested in collaborating or donating samples, they are probably the best indicators of where they might be in a disease. And if there is a family predisposition, or they think that they might be able to donate samples that help us understand some of these complexities of DM2 or DM1 or any of the other repeat expansion disorders, that helps us in general as a community just to understand what factors are important. So I echo what Chris said earlier, and just to jump on Darren's comment.
Okay. Well, thank you all. Thanks everyone for joining, and thank you to our esteemed panel for being with us today. We have a short break in the session. The next in the program, the next session starts at 11:30 Pacific. We will be talking with about NIH funding opportunities and looking at new data from the Marigold Foundation and listening a little bit to what the CDC is doing with their MD StarNet program. So please feel free to join, take a little bit of a break, and industry updates will follow the next session. So thank you all for being here at the conference. Thank you again to the fantastic speakers, and have a fantastic conference for the rest of the next 2 days.
Thank you so much.
Take
Thank
care.
you,
everyone.
Thanks, everybody. Goodbye.
Thank you.